Good. Okay, so I've called the talk Diatribes and Data, and we're going to start with the diatribes. So I'm going to warn you all before we start that we're about to enter an amyloid-free zone, and no one has ever been injured by this, but it may cause vertigo. So I just perhaps you would be well advised to fasten your seatbelts. No, of course, can't make it. There we go. So let's start. Okay, I have to prove that. So let's just start with the very simple question: what is Alzheimer's disease? And I think we can attribute the original thinking behind that, the answer to that question, to this man here, Alois Alzheimer, who had been the physician of record of this woman here, August Dieter, and he made many clinical observations during her lifetime, but when she passed away, he was very much engaged with the idea of the structure of the brain having a direct impact on its function. So he used what were then very modern silver staining techniques to stain the brain, and he saw these, what for him were abnormal deposits that he called amyloid plaques, and also a ghost-like appearance of a neuron that resembled what he called a neurofibrillary tangle or miliary tangle. And if we fast forward to the 1980s, the plaques were isolated by a pair of biochemists, Glenner and Wong, and they discovered that, in fact, the main peptide constituent was this small peptide that was cut from a larger protein. Actually, they just found that this small peptide, and later, or actually very soon after, the full parent peptide that generated A-beta was discovered, and it was found to be a large transmembrane protein called the amyloid precursor protein. So that led to the following idea, which very much grew out of Alzheimer's idea, that the structure of the brain was determinative of its function. That's why he felt the plaques were important. And so this idea led to what is called the amyloid cascade hypothesis, where this A-beta aggregates to form plaques. The plaques then trigger a series of events that lead to neurofibrillary tangles. These tangles and plaques together lead to the degeneration that we see on structural MRI of the brain of an Alzheimer's, and that overall this degenerative atrophy leads to the dementia, such as was seen in August Dieter. Now, this is a perfectly fine hypothesis, and in fact, for Alzheimer's Day, it was even very forward-looking and quite innovative. And over the years, it's been refined, and the most famous refining was done by John Hardy and Gerald Higgins, and this is their amyloid cascade hypothesis. It has seen many different iterations and variations, but the basic structure remains the same. Let's see if I can. I'm going to push my screen down for a moment, find my pointer. Well, no, I'm not. Yes, I am. Highlighter laser. Okay. So whether in familial AD or sporadic AD, that small A-beta peptide accumulates, begins to aggregate. The aggregates create an astrocytic and microglial response. There's a loss of ionic homeostasis and a loss of kinases unchained, leading to tau phosphorylation and tangles, widespread synaptic dysfunction and dementia. And as I say, the basic structure remains the same, even though there have been variations over the year. Well, this is a very fine hypothesis, and even in 1992, when it was first published, was forward-looking and clever. But as a mathematics friend of mine once said that the best thing about a good theory or a good hypothesis is you can take it out in the backyard and throw things at it and try and break it. Because a good hypothesis will stand up to testing. And it's in that testing that we gain more and more confidence about the hypothesis or discover additional data that leads us to say, well, we really need to rethink it or reformulate the hypothesis. So let's test the hypothesis. So the first thing we should do is we should look at people and say, well, okay, do they have amyloid in their brain? So if we, in the modern era, we can do this in living individuals because PET ligands have been developed that allow us to see in the living individual deposits of tau, which is the source of the neurofibrillary tangles, and deposits of amyloid, the source of the plaques. And what this slide shows, and it's you can find dozens of these on the internet if you want to go look, is that in a normal adult, the level of deposits is relatively low, whereas in these Alzheimer cases, the level of amyloid and tau deposition relatively high. So this, of course, is perfectly consistent with an amyloid cascade. But we run into a problem because we really have to do the control, and that is if we examine healthy seniors who are not cognitively impaired and ask, do they have any sort of level of amyloid deposits that we want to care about? The finding is that approximately 1/3 of them have plaques in their brains, and the density of those plaques would be consistent with a blind neuropathologist scoring it as Alzheimer's disease. Well, you may say, okay, that's fine, but maybe these people are just starting their Alzheimer's. Well, perhaps, but the risk of progressing to Alzheimer's is only modestly increased by the presence of these deposits. And indeed, if you stratify people based on their amyloid deposits, 5 years later, really half of them are still symptom-free. So in my world, that would at least cause me to look around for an alternate explanation between the dementia and linking the dementia and the amyloid deposits. But it gets even worse. Well, it's not that it gets even worse, but just to make that point as clearly as possible, you saw this slide before, but I hid the middle panel, which is in fact a normal older adult with cognition intact. And what you can see is that compared to the other normal adult, this individual had just as much or almost as much tau deposits as someone diagnosed with Alzheimer's. And they had, if anything, slightly more amyloid deposits than someone diagnosed with Alzheimer's. So by this observation, this deeply, deeply challenges one of the founding premises of the amyloid cascade hypothesis, because it's hard to imagine how you can have 1/3 of healthy individuals walking around with this level of amyloid and yet displaying virtually no cognitive symptoms. So let's test the amyloid hypothesis in the other direction. Let's take people who have the deposits and who have begun to show signs of dementia, and let's take the amyloid out of their brain. The prediction would be, if we're completely straightforward about it, is that this should either slow the progression of the disease or stop it altogether. Now, in neurodegenerative diseases, we're never looking to cure or restore function. No one's holding out that as a goal, but we should be able to slow or stop the disease. And in fact, it turns out we can remove amyloid from people's brains by any number of means. We can inhibit the proteases that are needed to generate the A beta peptide, and so you can't produce amyloid, so you can't produce plaques. And in what I like to say is an experiment that never should have been done, we discovered that you can actually vaccinate against amyloid, or you can give pre-engineered monoclonal antibodies, and we can get rid of it. And so let's look at what happens. And it's a very complicated slide. You don't have to look at the whole thing. But down this column are all the various clinical trials that have been run testing the strategy of removing amyloid, and starting either with mild to moderate Alzheimer's or prodromal Alzheimer's, or even what's called preclinical Alzheimer's. And you can see in this column, again, the details are not important, but all sorts of different strategies have been used to get rid of amyloid. And in this column here, you can see that most of them, not all of them, but most of them actually hit the target, meaning that by any number of criteria, the amyloid deposits in the brains of individuals in the trial, in the treatment arm, were reduced. And you can see from this column here, which charts the outcome, all those green boxes in this column indicate the trials in which people got better. And if you're saying to yourself, well, I don't see many green here, that's my point, which is that you can reduce amyloid and either have no change in cognitive status, or in many cases, cognition gets worse, and that's all the red ones. Now, we do have to pay attention to this trial here, which is the aducanumab story, and it's along with lecanemab and donanemab, we actually have to spend a little bit of time doing a deep dive into it, because as my reputation, which precedes me, might indicate, I have a pretty bad attitude about these responses as well. So the antibodies have been hailed as breakthroughs, but I don't think we can use that word. I think it's inappropriate. Aducanumab really did not meet its clinical endpoint. They had to jigger the analysis after it was halted for futility to actually end up with this significant sub-analysis. Lecanemab and donanemab, however, were clean successes. They both hit their target in reducing amyloid and also in improved cognition. All of them slowed decline, and by this I mean cognitive decline. And the figure that's cited is that between 25 and 35%, there was less decline in the treatment arm on monoclonals than there was on the placebo. And if you think about it, that means that all three of them were clear trials of the amyloid cascade hypothesis, and all three of them did what they were supposed to do. They lowered the PET ligand detection on CAT scans. But the effect sizes were incredibly small. So 25 to 35% is arithmetically correct, but in all the cases, that difference is less than 5% of the full cognitive scale that was being used, whether it's ADAS-Cog or the more fancy one that was used in the donanemab study. And in fact, this decline, this difference, which is graphed here, by studies that were done before these clinical trials were published, this level of difference would be predicted to be undetectable by the patient, by their caregiver, or by the physician. And what you can see is that in fact, the controls, that is the people on placebo, did indeed get worse. And it is true that lecanemab people got worse more slowly, but there's very little indication that this effect is increasing. And in fact, at this level, it's almost undetectable. This needs to be compared to what is now, standard of care. And this is the cholinesterase inhibitor donepezil. And the length of the trial that led to its approval is much shorter. And this, by the way, is 20 years ago. And it's shown here in this gray box. And what you can see is that the tracking for the placebo arm is almost identical. It's down to 1.5, and at the end, this is pretty close to 1.5. So even though there's a gap of 20 years between these two trials, the placebo arm is behaving as predicted. But look what happens on drug. So unlike the lecanemab story, with donepezil, people actually improve. And not only that, the difference between the treatment and placebo arm is double what it is. This treatment costs about $75 a month. This treatment costs about $3,500 a month. You make the call. And the risks are not small. There is up to a third of the people or even more had imaging abnormalities, the long-term consequences of which we don't know. We do know that there were deaths and that there were imaging abnormalities that we would be loath to take as benign. In addition to that, there were subgroup differences. Now, I'm going to list them for you, but I'm going to say right in the beginning that the studies were not powered for this to be statistically significant, but it is noteworthy. And noteworthy in particular is though women are more susceptible to Alzheimer's than men, men did 3 times better than women when treated with lecanemab. So that's sad. North Americans did twice as well as Europeans, which suggests there might have been trial site differences in the trial. And as is been very well recognized, ApoE4 carriers not only benefited very little, but their risk profile was much, much higher because they're far more susceptible to small hemorrhagic bleeds, which are called ARIA-H. And we don't need to go into that because I think we need to move on. And just as important for me as a basic scientist, I maintain we don't actually know why the treatments work or how they worked, and that's what this graph is. And I wanted to call it to your attention because I think it's a point that is relevant to all disease, but in particular to late onset diseases of aging. So if we look at our cognitive capacity over time, our cognitive capacity really peaks at middle age, and then it declines significantly over time. You will often see graphs where it's up here, but that's a lie. That's magical thinking. Our decline goes fairly steeply after middle age. What we do know, however, is that when dementia starts, that rate of decline accelerates. And I've graphed it here. It's just an illustration. There's no data backing this up. But this implication is important because what it says is that at any moment in time, someone with dementia is actually suffering from 2 losses. The first is the loss caused by the disease. In other words, the drop from someone with healthy aging and someone with dementia. But they're also suffering from what I call delta H, which is the normal and age-related decline. When we give a monoclonal antibody and we see the cognition improved, we have to ask ourselves, or at least I do, what are we affecting? Are we affecting the disease, which is the theory, or are we just slowing healthy aging? And I maintain we don't know that for the monoclonal antibodies. And I'm not done bitching, so hold on. Because I do want to talk about this slide. Now, for those of you in the Alzheimer field, you've probably seen this graph 100 times, not this particular graph, but the one it's based on. It is a wonderful graph stated as a hypothesis. And in fact, stated as a hypothesis, this graph is pretty much just a recapitulation of the Alzheimer cascade hypothesis of Hardy and Higgins that I showed you, which is to say, if we look at amyloid CSF, it increases. And after that, we begin to see amyloid on PET scans. That's followed by the appearance of tau, either in cerebrospinal fluid or using PET ligands. Then we begin to see brain shrinkage, and then we begin to see dementia. And this temporal order, however, while consistent with the idea that amyloid causes tau, causes brain shrinkage, causes dementia, it's certainly consistent with that idea, but it doesn't prove it unless we are already confident that we know the disease mechanism. And I hope by now I've convinced you that we don't. And if I haven't convinced you, see me after class and I will, or at least I'm going to try. And to underscore this point, we need to engage in a little bit of logic. So this is basically Alzheimer's hypothesis, which is that he saw plaques and tangles in the brain of August Dieter, and he saw that that was correlated with her dementia. And that's what this purple box is. And he hypothesized that it was the plaques and tangles that caused the dementia. And that's this black arrow here. And that was a perfectly valid hypothesis, as I said before. But if you think about it, and if you had the same data that Alzheimer had when he made that hypothesis, you could have equally well argued this direction, which is that it was the dementia that came first, and it led to the deposits of the plaques and tangles. Now, here's where the temporal order, A beta coming before dementia, makes this alternative less likely, but it doesn't disprove it completely. It certainly does make it unlikely. But I would maintain that, in fact, this is biology, and this kind of a simple arrangement is not really consistent with what we know the complicated physiology of our brains is. And in particular, what we don't know is that the correlation between plaques and tangles actually reflects the fact that both of them are susceptible to an outside factor, which I called X. The correlation is real, but it's not caused by any causality between these two. Rather, it is caused by an outside factor influencing both. So what are plausible X factors? Well, inflammation certainly is one, as is age. And what, of course, this requires is that inflammation is separately capable of causing plaques and tangles and also dementia. And we certainly know that this is true. Most people would agree that this is true. But we know that this is true because you experience it every time you get the flu. Your body reacts with a cytokine storm. And if I were to test your cognitive capacity while you were in bed with the flu, you would probably score very similarly to someone who had dementia. So I would argue that it's at least a plausible interpretation. And age as well. We know that age, plaques and tangles increase with age. We know dementia increases with age. So maybe there's just something about age that causes both of them, and that's why they're correlated. And just to be a bit, snarky, I would modify the graph to show that age also causes hair to turn gray. And by that standard, using the very same logic that Jack et al. Used in graphing in preparing this graph, by the very same logic, I could hypothesize that gray hair causes amyloid deposition, which causes tau deposition, et cetera, et cetera. I'm being ridiculous, of course. But my point is that logically, this is a hypothesis. This is not data. And you need to remember that. So we really have to do better. We need new ways of thinking. If I could wave a magic wand, the one thing that I would change more than any other is the definition of Alzheimer's. Basing it on amyloid just seems so ill-advised. Amyloid can certainly be part of the definition. I'm not arguing that may not be relevant. And I think we need to start thinking in terms of networks, not linear pathways. And so what do I mean by that? Well, let's think about all the many causes that could be contributing to Alzheimer's disease. And I've mentioned aging, and it's worth thinking that aging is actually the single greatest risk factor for Alzheimer's. And we really don't know much about its biology. I happen to like DNA damage. It's a driver of aging. It's linked to inflammation. I'm perfectly happy to include amyloid. I know this will shock some of you in the audience, but I am. It's okay. Its products are toxic, maybe. I'm happy to include tau as one of the causes. In fact, the biology backing up tau is much better than the biology backing up amyloid. But as I've said, inflammation certainly has to be near the top of anybody's list of a possible cause of Alzheimer's disease. I would add senescence, which increases with aging and actually may contribute to the inflammatory process. Vascular dementia is a very, very frequent comorbidity of Alzheimer's and probably contributes to the stress leading to it. Apolipoprotein E just recently has been described as having a new type of Alzheimer's driven by ApoE. Vesicle trafficking is hugely important. Calcium dynamics. I happen to like myelin, and we'll talk about that in a minute. Microbes. The pathogen theory of Alzheimer's is actually gaining an ascendancy. Sleep and circadian disruption. I mean, the list is endless, and to focus on just amyloid is, I think, counterproductive and is probably holding us back. So this is my way of ending this discussion by putting up what I call a simplified diagram of the aging brain. And you can see all of these various factors interact with one another and form a network. And it's this network that is responsible for the behavior and the changing behavior of the aging brain. And we can chart the emergence of dementia out of this network in any number of ways. If you're a Lois Alzheimer, and note that age really is at the center here. But if you're a Lois Alzheimer's, you can say, well, it's this APP cleavage that leads to beta amyloid plaques, and that's where the dementia comes from. All well and good. And in fact, if you're Hardy and Higgins, it's like, of course, because here, this is the amyloid cascade hypothesis right here. Age, APP cleavage, influenced by genetics, blocked by ApoE, A beta, also tau, and then you get dementia. But if you're me and you like oligodendrocytes and the loss of myelin integrity, you say, well, wait a minute, it's actually the myelin that's leading to the dementia. That's where it's coming from. And if you're one of the vascular people, you say, well, no. It's the hypoxia and ischemia that comes with vascular degeneracy, and that's where our dementia comes from. If you're into inflammation, you're going to say, no. It's the microglia and their, cytokine storm that is actually giving us the causes of dementia. And if you're into pathogens, that's where it comes from. And you know where I'm going with this, because this is actually Alzheimer's disease. And I think this is not a trivial point, because while in some ways it's a simple statement, what it says is that what we perceive clinically as Alzheimer's is actually a combination of underlying causes. And since age drives them all, they all contribute, and all of them help to shape the clinical symptoms. And I think this is a much more productive way of thinking about what Alzheimer's actually is. Okay, I am going to get down off my soapbox. That was the diatribe. So now, if you're up for it, we can do a little data. I'm willing to take any questions at this point, but I'm going to ask my host to monitor them for me, because I'm in presentation mode and I don't want to leave. So if there are none, we can save until the end when I stop sharing my screen. So let's move on to data. So let's start by asking the question that I hate most, because if the question is, if it isn't amyloid, then what is it? And as I think that network diagram tended to indicate, the answer is I'm not really sure. But again, let's be data-driven, and let's return to the idea of the genetics and the idea, that the amyloid precursor mutations in the amyloid precursor protein, or overexpression, as in Down syndrome, leads to early onset forms of Alzheimer's disease. And in fact, it was APP that was the first real genetic clue to the biochemistry and etiology of Alzheimer's. It's one of only three such genes that have been discovered. And I think with all the work that's gone into the genetics of AD, we're not likely to find any more. All three of them are autosomal dominants. And so what is APP? Well, it's a transmembrane protein that can be cleaved by shedases or secretases, as they're called. And this is a typical picture. It diagrams how you get with a beta secretase and a gamma secretase cleavage to the senile plaque. But what I want to point out is that this is a totally distorted view of what APP actually looks like. So it emphasizes a beta peptide and totally distorts both the N and C terminus. And I'd like to argue that this is a much more representative image of what APP actually looks like. And A beta, this little dooha here, is really only about 5 to 6% of the total sequence. The N terminus, which sticks outside or into the lumen of a vesicle, is quite large, as shown here, and its structure is very reminiscent of a cell adhesion or a cell signaling molecule. And that idea is bolstered by the fact that it's capable of forming dimers with itself by dimerizing it in internal domains, or with other APP molecules, or with APP family members known as APLP1 or APLP2. We know from some beautiful work that Wei Zhang has done at Baylor that the interaction is crucial for synaptogenesis and the formation of the neuromuscular junction. So, and not only that, the C terminus, although it is short, is just chock-a-block full of interacting motifs. So there is this YNPTY, which is interacting with GRAB2, with FE65, with DAB1, which puts it in the reelin pathway. It's got a huge domain here, which interacts with kinesin and dynein, so it's going to be involved with vesicle trafficking. It's a very rich protein, and to think of it just as the precursor to amyloid is okay. It's silly. So that's where my postdoc, Jack Ma, Chinese name Fulin, got into the work. And since we've moved to Pitt, he's been collaborating with Himanshu Alkakar. And I want to tell you about some of their findings. So what Jack discovered is that, and this was known to a certain extent before, but APP is a very, very sensitive protein, and it reacts very quickly to changes in the neuron. And in fact, just a simple increase in neuronal activity will cause its levels to rise dramatically. So after 24 hours of just 1 micromolar glutamate, which is a relatively low concentration, you can see the immunohistochemistry for APP goes way up, and that's illustrated here, quantified there. It's activity dependent because if you add an NMDA blocker like DCPP, you find that you can completely block that. If we look at the level of message, the response is extraordinarily rapid. So we hit him with glutamate at time 0, and within an hour, the level of message has gone up 3-fold. So almost an early response gene kind of response. One of the things that happens when APP levels go up is that the APP protein begins to invade the axon initial segment. So I am sure that all our neuroscientists in the audience know that the axon initial segment is where action potentials are initiated. So it's an incredibly important part of the neuron. When APP invades, 3 things happen. First of all, its levels go up. This is the intensity of APP within the initial segment. The AIS, or axon initial segment itself, becomes shorter. and it moves away from the cell body. And again, both the shortening and the shifting are both blocked by the NMDA receptor antagonist. So all of these changes would be predicted to lower the firing probability of a neuron. And without going into the biophysics of it, it's because the current is, it attenuates faster in an axon if the initial segment is moved further away. And if it's shorter, there's not room for as many channels, and so the channel conductance will also go down. The details aren't important, but the point is that activity induces APP, which then will quiet neuronal activity. I want you to start thinking about this as a sort of a governor. That is to say, APP is in a position to actually prevent a neuron from overheating. Now, nothing I've said about that so far would relate to Alzheimer's disease directly, but I want to throw out this little bit of candy, which is that you don't need glutamate as a way of increasing APP. You can just transfect APP, and that alone is enough to move the AIS and decrease neuronal activity. I'll show you the activity in a minute. But what's interesting is that wild-type APP is very effective, but APPs that carry familial Alzheimer mutations are even more effective. And that can be seen most clearly in the length of the AIS, but it's also quite clear in the shift, although in this case, it wasn't significant. I know you're thinking that it must be A beta, but I said this is an amyloid-free zone. So I'm sorry, when you apply exogenous A beta by itself, it has no effect. Either fibrillar forms of A beta or oligomeric forms, doesn't matter. Neither the length of the AIS nor its distance from the cell body changes. So it's not A beta, but it's as important in vivo as it is in vivo. So in both human and mice, in conditions where APP is upregulated, the axon initial segment shortens. So this is a human tissue stained with ankyrin G to show you the initial segment, and APP to show you normally that there are very low levels of APP even in the aging brain, although there's more than in the young brain. But in the Alzheimer case, there's a lot of APP found. It's interesting that a lot of it is intracellular. And when you look at the AIS, the initial segment, they're considerably shorter, even though their density is not changed. APP physically associates with the scaffolding proteins of the AIS. It's a complicated slide, but let me just say that if you blot with the AIS scaffolding protein, ankyrin G, and then blot the pull downs from ankyrin G itself, so it's a co-blot, it works. Beta-4 spectrin, we know is part of the scaffold that ankyrin G is a part of. You can blot with that, but note that you can pull down with any number of APP antibodies and also find the ankyrin G being physically associated with APP. And that association and that invasion of the initial segment has the expected effect on activity. So this is GCAMP that has been transfected into neurons, and here it's been transfected along with APP, and you can see that the spontaneous activity is dramatically reduced, both in terms of the intensity and also in terms of the frequency. We can go to multi-electrode arrays and make that even more precise. So this is where Himanshu comes into the picture. We use a very high density array, which basically uses CMOS technology to measure potentials on electrodes that are spaced just 17 microns apart. Each of these squares is a separate electrode. And we can define hotspots, which you can see these hotspots overlay structures that we see immunologically as MAP2 positive cells, so they're neurons. And what we find that is interesting is that using these multi-electrode arrays and now to stimulate the cell, if the cell is normally will respond to voltages, millivolt stimulation between 20 and 40, if that neuron has been transfected 1 week earlier with APP, you see that it takes much more current or much more voltage to stimulate the same spike probability. So this overexpression of APP dampens neuronal activity, and that's at the level of the single cell, but you can also see it at the level of the network. So what this is it's a plot of 1,000 of those electrodes selected by their activity. This dimension actually has no anatomical meaning. It's basically just a list of electrodes. And what you can see is that at 2 weeks, the cultures begin to mature. And when they do, there's a lot of synchronous bursting that goes on. That becomes even stronger at 4 days later. And now you begin to see increased numbers of interburst activity. That interburst activity increases until by 26 days, you have a very rich neuronal network that's going on. If you look instead at a wild type neuron, I've just reproduced those images here, but now look at neurons from a mouse model of Alzheimer's that just overexpresses the APP gene, you can see that the maturation of the network is dramatically slowed. So not only are the individual cells affected, but the entire behavior of the network is affected in terms of its maturation. So I would wrap this part up by saying that APP has a life outside of amyloid. It increases very rapidly with neuronal activity. It almost like an immediate early response. It alters the structure of the axon initial segment, and in doing so, reduces the probability of the firing. I'm going to predict, I have no data yet, but that it's the C-terminal interaction with the cytoskeleton that I showed you that on the diagram that is crucial. But in point of fact, if we think about it functionally, what that means is that APP is acting as a governor, protecting neurons from overheating and from the ill effects of hyperactivity. And it's a little bit counterintuitive because for those of you who know the Alzheimer field, there's increasing evidence that Alzheimer's is associated with increased epileptic activity. And that doesn't fit directly with this idea that APP quiets the neuron. But what I would argue is that we don't know yet, and that should come up as a question later, so I'll plant it now, whether the effect is similar in the APP effect is similar with excitatory and inhibitory neurons. And all it would take would be a slight quantitative enhancement in the inhibitory neurons to give you exactly the change in the EI balance of the network that would lead to epilepsy. That's speculation in the absence of data, but it's my seminar, so I get to do that. But I want to move on because not only does the amyloid precursor protein as a whole impact activity, it gets involved as a cell adhesion molecule. And this is the work now that Jack did, but he did it in conjunction with a former postdoc of mine who's currently on the faculty at Polytechnic University in Hong Kong. And they are involved in a myelin story, which looks at the role of oligodendrocytes in the etiology of Alzheimer's disease. So myelin is very attractive as a possible driver of Alzheimer's disease. And in part, that's because the quantity of myelin in the brain is huge in humans, and it's vastly increased compared to other creatures. It correlates very well with cognition. And in fact, that would imply, because I showed you that graph before, that myelin loss occurs with age and its loss accelerates in Alzheimer's. And let me just show you what that data looks like. This is data from George Bartzokas, which I've replotted. George Bartzokas was really the first one to really push the myelin hypothesis. And what he showed is that this is the normal age-related loss, and that people with Alzheimer's, in fact, deteriorate even more rapidly. what Frankie then was able to do was to show that whether you look at the total oligodendrocyte population, which he marked with OLIG2, a transcription factor, or look at the oligos that are actively myelinating, which is the second transcription factor known as MYRF, you find that there is a vast decrease in the number of oligos and in the number of actively myelinating oligos in Alzheimer's disease. Interestingly, even in individuals who have dementia but no amyloid plaque deposits, you still see a reduction in. So it's correlating with dementia, not necessarily with the presence of plaques. But I'm going to hypothesize that APP plays a role here as well, because this is a diagram of what a myelin wrapping of an axon looks like. And I'm going to propose that as a cell adhesion molecule, it actually modulates the interaction between the neuronal axon and the wraps of oligodendrocytes. And if you're following along, I hope you're saying, whoa, 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 whoa. There are no plaques in the white matter until very late in Alzheimer's disease. Do oligodendrocytes even make APP? And I would say, well, I'm glad you asked that question. Because if you go to the Human Protein Atlas and you look up APP and you sort brain expression, this is now single-cell RNA-seq data, and you sort brain expression based on, you sort the data based on expression levels, what you'll find is that all the cells of the brain express very high levels of APP. I mean, these are numbers that are quite significant in terms of reads per million. But it is noteworthy that the cells type that makes the most APP of all the cells in the brain are the oligodendrocytes. So, okay, they make APP, but now what about the secretases? Well, that's even worse, or better. Alpha secretase, beta secretase, gamma secretase. In all three cases, the cell type that has the most message for these three proteases is the oligodendrocyte. So they're perfectly capable of not only making APP and functioning as the interacting protein, but they're also capable of making A beta. And you would say, but wait a minute, I mean, A beta, those are plaques that come in the brain. Is there any amyloid in the white matter? Well, actually, there might be. So again, I just pulled this off the web. You can pull any number of images just like this off the web. This is an image of florbetapir in a living individual, one who is cognitively normal and one who is having just the beginnings of dementia, mild cognitive impairment. And this is a horizontal, this is a coronal section, a horizontal section, and a sagittal section. And I don't know how many of you have had neuroanatomy, but what's stained? Well, does anyone know what this is? If you answered corpus callosum, you would be correct. And so even when you get to the more advanced cognition, the deposits of whatever this florbetapir ligand is binding to are, I hate to say, primarily in the white matter. So there's no question that there are no plaques there, but I think the imaging data is beginning to suggest that there may just be something going on, that this binding has been dismissed as an artifact, but I would maintain we need to reconsider just how artifactual it really is. And just to drive that point home, let's do some immunocytochemistry where we double label with APP and myelin basic protein, MAP2 for neurons and DAPI for nuclei. And here is from the corpus callosum of that Alzheimer mouse model that I showed you before. And look at this. All the yellow that you see here is the co-localization of APP with myelin basic protein. And confocal images clearly indicate that APP is located in stuff that looks like myelin. Here's from the white matter, subcortical white matter of the human Alzheimer case. And same thing, you see that there is quite a bit of yellow overlap where the presence of APP overlaps with that of the myelin basic protein. And just to blow it up and look at it up close and personal, as they say, APP in red, myelin basic protein in green. This is from an example in the human. This is an example in the mouse. And what you can see is that the axon is relatively clear. If you turn up the gain, you can in fact see APP there. But what you notice is that there's a lot of yellow where APP and myelin overlap. And even where they don't quite overlap, the location based on the myelin basic protein staining makes it very clear that it's not in the axon or even in the membrane of the neuron. Rather, it's probably in the myelin. And to underline that this is regulating the interaction between oligodendrocytes and the neuronal axon, let's turn to one of the most sensitive interaction points between the two cell types, and that's the node of Ranvier. So again, not to dwell on the details, but the myelin wrapping of neuronal axons is not continuous. It's interrupted by small 1 or 2 micron gaps with a very high density of channels. And by the way, the structure of those nodes is virtually identical to the structure of the axon initial segment. So it will not surprise you that in the R140 mouse and also in human Alzheimer's disease, in the node of Ranvier, the level of APP goes up dramatically. And that has consequences because in both the mouse and in the human, the node actually increases in width. So as they fill with amyloid precursor protein, the length of the node increases. This is in the mouse. Here it is done graphically in the human. In both cases, the paranode, and again, I don't want to go into the biology, the paranodes shrink. So in fact, the total structure doesn't actually change that much, but the node itself increases. And by the way, expanding the node, physiological experiments have shown that this will decrease conduction velocity and it will also increase failure. So once again, APP is actually serving as a governor. It's quieting a neuron down, quieting its transmission. And this is very recent data, but we asked what is the effect, the acute effect of APP overexpressing. So we both overexpressed it by injecting lentiviruses into a normal brain, a wild-type mouse brain that overexpressed APP. And as you can see, this is the uninjected brain. Myelin basic protein is in green. And when you overexpress APP, its levels go down. By contrast, this is the uninjected brain stained with PLP. I've lowered the exposure time to make a point, which is that when you inject an shRNA to knock down the level of APP, the expression of myelin basic protein goes up. So what else can APP do? Well, it looks like not only is it acting to affect the physiology of the neuron, but it also looks like it's affecting the cell-cell interaction between neurons and oligodendrocytes. And the argument is that this elevated level of APP, such as you find with age, with Alzheimer's disease, and with excessive neuronal activity, actually impairs the interaction. The node expands, myelin protein synthesis declines, and once again, we have APP acting as a neuroprotection device in neurons. And I would argue that normally these effects that I just showed you are very dynamic and very reversible. They're part of all of our normal physiologies. The problem comes with age. And with age, and even worse with Alzheimer's disease, the elevation in APP becomes chronic and ultimately becomes irreversible. And this leads to changes in the network function, such as those that I showed you with the multi-electrode arrays. It leads to myelin deficiencies, and these contribute directly to the symptoms of dementia. Both of these contribute to the symptoms of dementia. Thinking about it therapeutically, I would argue that what you really want to focus on is not the oligo itself, not the neuron itself, but rather that the target of dementia is the interaction between the two. The failure of this interaction can begin in the oligo or it can begin in the neuron, but it's the interaction that matters, so it doesn't really matter where it starts. Alzheimer's actually represents a vicious cycle of failures in this interaction and in other activities. And just to beat that dead horse one more time, because I don't think the horse is dead yet, you don't need A beta to explain the genetics of amyloid precursor protein. And I hope that's been a subtext of what I've said, but I want to make it explicit right now. All of these studies looking at alternate roles of APP, really the genetics of familial Alzheimer's disease do not identify A beta. They identify APP and its processing. I've got some ideas about what presenilin does, but the hour is late and I need to stop. So that'll be for next time. So I want to come back to this slide because I think more than anything, this captures my view of what Alzheimer's dementia looks like, that it is a multisystemic combination of insults to the brain that ultimately disrupt the normal neuronal function, neuronal network function, and lead to the symptoms of dementia. And so with that, I just once again want to thank so many people who have helped with this work, and I probably have not listed them all, and the generous support. Actually, this work began in Hong Kong, so my wonderful HKUST helped out, as did the Hong Kong Research Grants Council, and then Pitt has been helpful, and now, despite it all, I've been able to go back and get refunded from the NIH. So with that, I thank you for your attention, and I'll take questions.